Thank you, Dominic, for the introduction, and thank you also, Bill, to both of you also for the invitation. It's a great pleasure to present at this series. As you briefly mentioned, I will be speaking about one of the main topics of my lab, which is the development of new therapeutic strategies for muscle diseases, mostly for muscular dystrophies, using iPS cells and myogenic cells derived from iPS cells, and also more recent technologies that we developed in the lab based upon the generation of three-dimensional artificial muscles, mostly for disease modeling, but also to have some complex therapies for complex platform for therapy development. As Dominic briefly mentioned already, my lab is at UCL, and from just a few months ago, actually just before the lockdown, we also have another attached lab at the Crick. So that's also another very exciting opportunity to foster new collaborations over there. Let me see if I can. Yeah. So these three thematic areas that you can see in this slide are actually the three main areas that we work on in the lab. So we really focus on studying muscle regeneration and muscle stem cells, and how we can improve this process for incurable neuromuscular diseases. My background personally is in gene and cell therapy for muscle diseases, and that's where I've been working. This is what I was doing during my PhD and during my postdoc. And then initially, I was mostly focusing on that, but we're still doing quite a lot of work on this area. On the other hand, use the sort of same technologies that we developed over the years for muscle gene and cell therapy, also in other areas such as tissue engineering and disease modeling. And actually, this is what I will be focusing today. I mean, for the sake of time, I won't be able to give an overview of the sort of three areas that we continue to work on. I will be focusing more on the sort of boundary of these two areas, the tissue engineering and disease modeling using iPS cells. You know, whoever is interested can have a look at our website or can drop me an email and we can have a chat about other projects going on gene and cell therapy. So as you might have seen from the title already, I will obviously be talking about skeletal muscle. So just a couple of words for the non-muscle experts. Muscle, it is the most abundant human tissue, and it is frequently forgotten, and actually it is the main output of the central nervous system as well. So pretty much anything coming out of the nervous system goes to a muscle eventually. It has a complex structure and functions as well. As you can see in this cartoon, muscle fibers live in a very complex environment surrounded by blood vessels, by motor neurons, and by these little cells here underneath this layer of connective tissue, this basal lamina here, called the epimysium, the endomysium of the muscle fiber. And these little cells live underneath, called satellite cells, and they're actually the key muscle stem cell population sustaining the regeneration of skeletal muscle. However, this regenerative process is not optimal, and indeed, for this and other reasons, unfortunately, muscle is also the target of extremely severe disorders, of which muscular dystrophies are one of the key examples. And most of them, unfortunately, if not all of them actually, but the vast majority of them, are quite severe and incurable at the moment. Very severe actually and incurable. So there is really a desperate need to understand disease mechanisms and as a consequence of that, also develop therapies for these terrible conditions. So to this aim, we've been focusing on trying to develop high-fidelity platforms to model muscular dystrophy in a dish, so that we can also understand a bit better what's going on in these diseases and try to develop therapies for to cure them. So to model muscular dystrophies, we actually want to have models that are patient-specific, that are time-resolved, i.e. giving us an idea of what's going on at different stages of muscle differentiation, and also will be, and models that can give us a possibility to dissect molecular pathways. And to tackle all these issues, one of the key solutions we've been working on is reprogramming cells to iPS cells and then directing their differentiation into muscle progenitors and myotubes and myofibers in vitro. And we've been pioneering this for muscular dystrophy patients a number of years ago, as you can see in this paper here, where we also did genetic correction and transplantation of the same cells. Later on, we also expanded this area by developing protocols for the large-scale expansion of inducible myogenic cells from iPS cells. And more recently, and I will be talking a bit more about this, we've also been able to use those iPS-derived myogenic cells that I just mentioned, and by merging them with biomaterials and different differentiation protocols, we've been also able to create complex artificial muscles, three-dimensional artificial muscles of muscular dystrophies, which can recapitulate key phenotypes in vitro, as you will see in a few slides. So before getting started, just a few words about making skeletal muscle from human iPS cells. This is a slide that I've adapted from a paper of Olivier Pourquier published recently. But essentially, the point I would like to make here is that there are different ways to go from a pluripotent cell to a muscle fiber. The idea is that there are two main routes to do so. One of the first routes explored and still actually actively used, including in my lab, is to use transgene-based platforms to force, in a way, the differentiation of iPS cells into myogenic cells. There are different ways to do that. There are different transcription factors that can be expressed at specific time points of the iPS cell or pluripotent cell differentiation to induce the myogenic program, the skeletal myogenic program, PAX3 or PAX7, like the work done by Rita Berlinguero, for example, or MyoD, as we did years ago. And more recently, the focus has been redirected into using small molecule mediated differentiation strategies, which are transgene-free by definition and have the advantage in a way to recapitulate myogenesis with higher fidelity when one wants to also see what happens at early stages, for example, before you induce the overexpression of one of these transcription factors, or for example, when one wants to keep a better pool of progenitors, undifferentiated progenitors near your muscle fibers, which is sometimes difficult to do when the specific transcription factors are expressed. Bottom line is that depending on your need, you can go from one option to the other. For example, one of the advantages of the transgene-based differentiation is that they are synchronous and they are sometimes a slightly higher efficiency, slightly higher reproducibility, or for example, less contaminants in your population. Whereas the small molecule mediated strategies tend to be more high fidelity in terms of recapitulating developmental events and muscle histogenesis. So we, as I said, wanted to embark into this project of modeling muscle disorder using iPS-derived myogenic progenitors. After we established this platform mostly for gene and cell therapy, we sort of redirected our attention into this area because in a way, it could also give us a chance to have a fast track of, for example, currently available compounds for clinical application. And one of the first target diseases we decided to work on is this group of diseases called skeletal muscle laminopathies. They're also known as LMNA-related muscular dystrophies. Essentially, these are muscular dystrophies. You can see them here: Emery-Dreifuss muscular dystrophy, limb-girdle muscular dystrophy 1B, and of particular interest for us, congenital muscular dystrophies, which are all caused by mutation in lamin C, this LMNA gene, which is a nuclear envelope protein, gene coding for a nuclear envelope protein, which essentially confers stability to the cell nucleus and also regulates gene expression and chromatin organization. And when mutated, it causes, apart from this group of muscular dystrophies that I mentioned, a number of other diseases called laminopathies. Laminopathies because essentially, as I said, this protein lives in the nuclear lamina. And at the end of the day, it gives you a dysfunctional nuclear envelope. For example, progeria, the premature aging syndrome, it's a famous example of the diseases caused by mutation in lamin AC. So then we wanted to embark into this project to study the pathophysiology of this group of diseases, the skeletal muscle laminopathies, which is still poorly understood, and try to clarify better the genotype-phenotype correlation, which is again quite poor in these diseases, but eventually also to develop therapies. So this is an interesting story because it started a number of years ago in collaboration and is still being developed in tight collaboration with my friend and colleague Pete Zammit at King's College. And it was started in our lab, in my lab and Pete Zamm, by a talented PhD student, Heather Steele-Stallard, you see her here, who was a student from the BBSRC LIDO program. So what Heather started to do, and then Luca Pinton followed up in the lab at present, and also Dan, more recently Dan Moore here, and you will see data for all the three of them in the next few slides. What Heather initially wanted to do was to try to see whether mutations in lamin AC, and particularly these are the first three mutations that we started to work on, I should say in LMNA-mutant iPS cells more than lamin AC, mutations of those iPS cells would result into nuclear shape abnormality upon differentiation. Now, nuclear shape abnormalities are a key phenotypic hallmark of this group of diseases in vitro. So as I said, this gene has a key role for nuclear integrity and nuclear envelope structural integrity. Hence, when absent or mutated, or when acting as a dominant negative, because these are dominant diseases in particular, you end up having a dysmorphic nucleus. And we were quite pleased to see that just at the beginning of the muscle differentiation protocol, we could already identify a number of dysmorphic nuclei, as you can see in this panel here, in all the patients that we differentiated, in all the iPS cells that we differentiated from those patients. And the number you see here is the nuclear circularity ratio, which will be clearer in a couple of slides. So that was quite exciting. So we were starting to see something interesting. And then we moved on, and we not only observed these nuclear shape abnormalities in those differentiating iPS cells, but we also observed mislocalization of the nuclear envelope proteins. So I mentioned about lamin AC earlier on. For time reasons, I won't be able to detail all the other proteins that interact with lamin AC, but I put here some other examples, such as hemerin and lamin B1. And in control cells, you see that these proteins are nicely distributed around the cell nucleus, like a rim, as you can see here, or in these confocal images. But in our three patients that we kindly received from iPS that we kindly received from Cure CMD and Cellular Dynamics in the US, you can see the lamin AC is actually mislocalized. You don't have a nice and even ring of lamin AC around the nuclear envelope, but there are different abnormal structures there, such as aggregates of lamin AC, for example, this one, honeycomb formation, foci, cupping at specific parts of the nuclei, not only of lamin AC, but also of these other nuclear envelope proteins. And these are quantified here. So again, to summarize this part so far, essentially we were able to see abnormal morphology of the nuclei and abnormal localization of lamin AC just halfway through the differentiation towards the mesodermal lineage without even reaching the myogenic state. So going back to the nuclear morphology and the nuclear morphology, nuclear abnormalities, we then, as I mentioned earlier, we started also to quantify them, and we quantify them in proliferating cells. And you see a quantification here using the conturation, which I mentioned before. Essentially, the higher the number, so 1 is the highest you can get, the rounder the nucleus. The lower the number, the more dysmorphic is the nucleus. These are two controls. These are our three patients in the proliferating, in this stage, skeletal inducible myogenic cells from iPS cells of those three patients. And you can see that they were nicely statistically significant in terms of decrease of the nuclear contour ratio, meaning that we started to have also quantifiable objective phenotype there. And this is also quantified in a different way here, looking at the specific types of abnormality. again, short story is that we have a nuclear morphology abnormality in the skeletal myogenic cells derived from iPS cells, and we can quantify that. What was interesting, though, is that the moment you then move on and try to see what happens in the fully differentiated iPS cells, when I say fully differentiated, I refer to the actual skeletal myotubes. When we say myotubes in the skeletal muscle field, we refer to muscle fibers in vitro, multinucleated muscle fibers in vitro. When we were looking at the phenotype of those nuclei in vitro, we observed something interesting. So we were expecting initially to see worsening of this contour ratio, basically seeing even a lower number here in the myotubes, because that is where the disease should manifest in the actual myofibers. However, this graph is a bit complex, but again, to cut another long story short, if you look at the myotube nuclei, so the red bit, just in one patient now, in one patient out of 3, we had some statistically significant nuclear morphology alteration. So rather than getting worse, this phenotype was getting better in the myotubes. So that was a bit of a surprise for us, and we started to wonder whether that could be caused by limitation of the standard monolayer culture that we were using to model this phenotype in vitro. This is just simple differentiation on a monolayer of myofibers. And you know, muscle is a tissue which is highly dynamic, is subjected to tension. There is a complex three-dimensional niche, as I said in one of the first few slides. So we wanted to have a better system to recapitulate the three-dimensional muscle structure and the forces applied to muscle to see whether those will have a different, will give us a different outcome on the nuclear morphology, closer to the real life scenario. And luckily for us, at that time, we had another project going on in the lab, initially developed by another very talented PhD student, Sara Maffioletti. And we were basically with Sara trying to develop a three-dimensional muscle model, fully developed from iPS cells, essentially from a single cell, differentiating those iPS cells into different lineages. The skeletal muscle lineage is something we were already doing routinely and we were good at, but then we got some experience in getting endothelial cells out of the same patients, and then perivascular cells such as pericytes, and also later on motor neurons. And we started to fuse them all together with different biomaterials and obtained what you will see in a minute, essentially a little three-dimensional skeletal muscle construct built on the platform initially developed by Thomas Eschenhagen, who was a tight collaboration of this project in Germany for cardiac tissue engineering. And the idea behind this was then to have a platform essentially to do what I mentioned before, to do high fidelity disease modeling, but also to potentially use it for tissue replacement as a regenerative medicine product, also to screen not only drugs, but also gene and cell therapy products. And as I said, this is the project that was started by Sara Maffioletti and published a couple of years ago now in Cell Reports, and then completed by Shipita Sarkar, the postdoc in the lab. And it's mainly funded by an European Research Council grant. So we were, at this stage, we started to think, well, we might have the ideal platform then to test the hypothesis that those nuclear abnormalities that I mentioned earlier in the laminopathies may be exacerbated in a three-dimensional environment. And this is just to give you a brief overview of the sort of strategy that we use. These are the sort of iPS cells that we use initially to build up the three-dimensional platform. I'm going to spend a couple of slides just to describe you the 3D platform, and then I will be focusing again on the laminopathy phenotype. This is what we see in 2D. And then, as I said, we take our protocol, but also another transgene-free protocol to validate the entire strategy. And this is the transgene-free protocol that we have utilized. Fuse them with the platform developed by Thomas Eschenhagen, and this is what we get in 3D. This is the central bit of that little strip of muscle that you've seen in the previous slide. And there is abundant myosin heavy chain, which is here shown in red, and as the name says, is a key marker of terminal myogenic differentiation. This is just a blow up of one of those previous images where you can see healthy a three-dimensional iPS-derived artificial muscle, and another muscle derived from a dystrophic iPS line. And this is a zoom in to show you the alignment after about less than 10 days of differentiation, actually. Of course, we're far away from the morphology of a normal adult muscle, and that was never our aim, and it's unreasonable to do that within 10 days or so. But you can see that there is a beautiful alignment, multinucleation, and abundant myosin heavy chain production. And these are just additional characterization of those constructs that we did, and you can, for the sake of time, refer to the paper to get a bit more information. This is, again, also to give you an idea of how these constructs look like. This is light sheet microscopy done on one of those 3D constructs. You don't see the internal fibers because essentially the reconstruction is only on the external layers, but you can see that there are abundant myosin. Actually, here you have titin, which is even a later marker of myogenic differentiation. And you also see dystrophy, which is another late marker of myogenic differentiation, starting to be expressed by around day 7 of differentiation, mostly at the poles of this 3D constructs. We did quite a lot of other experiments to look at maturation of those artificial muscles, looking at sarcomeric actin. As you can see here, these nice little fibers full of sarcomeric actin with multiple nucleation inside in transverse section. There are kind of rudimental sarcomeres already formed around day 7. And we can also see PAC7 positive satellite-like cells when we develop these muscles using a transgene-free protocol, and which was quite exciting for us to see, because we can also then recreate not only the myofibers, but also a niche permissive to the establishment of a pool of stem cells in these muscles. And PAC7 is the key marker of those muscle stem cells, satellite cells that I mentioned before. And the muscles were able also to respond with spikes of calcium transient upon caffeine administration. So short story here is so far so good, quite exciting. We were getting something which looked like a muscle. We could also implant those muscles to check if they were biocompatible, and we did it in immune deficient mice. And this is the implant after a couple of weeks, as you can see here, vascularized microscopically and microscopically. You see now the capillaries in between the human nuclei. Now we're using laminate-C to look at the human nuclei, not to mark the disease nuclei. So don't get confused by saying laminate-C over here. And iselectin here also marks the blood vessel cells inside those implants. Essentially, this is a compound that is given intravenously to the mice. And if you see it in your construct, it unequivocally says that your construct is vascularized. And we also look at forced recovery of mice implanted with this construct, and we excitingly realized that we were getting a much better recovery when the implants were multi-lineage, as in having not only the myofibers inside, but also other isogenic cell types, such as the endothelial cells and the pericytes. And this will be clearer towards the end of my talk. And indeed, actually, I'll spend a few more words about that just now. And indeed, what we did was to add, as I said earlier, not only myogenic cells to our 3D construct, but also additional cell types to support them, such as vascular cells derived using Christine Mammery's protocol. And we obtained both endothelial cells, which are here shown by CD31 positive staining, and pericytes shown here by SM22 positive staining. We combine them with isogenic myofibers in vivo, and this is what we got. You can see the blood vessel cells growing next to the myofibers here, and also the pericytes highlighted here with a GFP mark of the assorted cells next to endothelial cells and next to the myofibers. And then we also moved this platform further on and added not only vascular cells, but also motor neurons, which are of course critical muscle function. And this is work we done in collaboration with Ricky Patani at the Crick, and also using alternative protocols as well. But this is the example you see above here is the protocol that was developed in Ricky's lab. And you can see we had a nice pure population of motor neurons that we added to our muscle constructs next to the pericytes, the endothelial cells, and the myofibers, having a quadruple lineage, isogenic, human, fully iPS-derived, three-dimensional artificial muscle. And these are all the different cell types highlighted in different colors in this confocal image. And you can see also the generation. I don't want to say neuromuscular junctions because we don't have hardcore proof that those are neuromuscular junctions, but we can clearly see the presence of bungarotoxin positive clusters. Bungarotoxin binds the neuromuscular to the postsynaptic neuromuscular area on the myofibers. You can see them here, and you can see that that area in this other example here is juxtaposed to the termination of an SMI32 positive motor neuron. And interestingly, as you can see in this example here, the fibers in these 3D muscles, which are, I won't say innervated, but at least that show the presence of bungarotoxin positive patches, are also show the presence of much nicer striation, suggesting, as it should be the case, that the innervation might also facilitate maturation. Now, after this long introduction on our three-dimensional muscle platform, basically we took those 3 iPS lines from the laminopathy patients that I told you towards the beginning of my talk, and put them through this pipeline. So we basically generated artificial muscle using those patients, and we were quite excited to observe that at variance with the monolayer, which you see here in some examples on the left side of this panel, we had a much different nuclear morphology in the three-dimensional reconstruction of the nuclei in the three-dimensional artificial muscle. So the nuclei now, rather than looking like pancakes, as you can see here on the left in the monolayer, they are much more rounded. And interestingly enough, as you can see already here, they are very elongated. And if you try to quantify this, on the top graph, you see just the quantification of the number of nuclear abnormalities. But more interestingly, on the bottom graph, you see the nuclear elongation as a single phenotypic readout that we've been using there, which was relatively poorly described in this group of diseases. But we were quite excited to see that, because this is now another objective outcome measure, an objective phenotypic readout that we can get in our 3D muscles. And that was quite statistically significant in all the examples that we have seen. And this is in keeping with what observed in LMNA-mutant mouse cardiomyocytes in vivo, and also in some LMNA-mutant human primary myoblasts in some 3D cultures in vitro. So telling us that the model that we developed is quite high fidelity in terms of mimicking what's actually happening in real life. Then we went on, and specifically Heather here did a number of other investigations together with Luca Pintone in the lab. They've been looking at not only at the morphology of those nuclei, but also detailing the difference between the monolayer and the artificial muscle on a mutation-specific basis, as you can see in these graphs, both looking at the overall number of abnormally shaped nuclei and at the nuclear length, comparing the monolayer and the artificial muscle on a mutation-specific basis. And as you can see, in the vast majority of the cases, the 3D model gives you a high pickup rate of the abnormalities. You can notice that here, and also for the elongation here and here, for example. So that was quite interesting for us, because it's actually proving what we were suspecting from the very beginning, that a system which will mimic the three-dimensional niche of muscle and the forces applied to muscle, because those 3D constructs under passive tension will unravel more nuclear abnormalities, because those nuclei are now challenged by those stimuli. And these are videos that we also generated later on when we perfected the staining protocol, also to stain not only the nuclei in the 3D muscle, but also the actual fibers together. And you can see that this is an example showing titin, which is a late muscle differentiation marker that you can see in the cytoplasm of this muscle fiber to make it easy. And this is lamin AC. Now the colors are opposite of what you've seen before. This is a normal wild type artificial muscle. So you can see the nuclei inside the fiber. You can see that the nuclei are aligned along the direction of the fibers, and they are slightly elongated as you would expect in nuclei, but nothing particularly abnormal. And then when you try to do the same thing with the LMNA-mutant iPSC-derived artificial muscles, oops, something is, let's see. Yeah, so you can actually see now that the nuclei are massively elongated. For example, look at this one here. It's coming back now there. Massively elongated and quite dysmorphic as well. There are less dysmorphic nuclei in this field, but much more elongated ones, suggesting that elongation could probably be a very good and reliable phenotypic readout for future drug screening projects. So then more recently, and this is a recent unpublished work that we've been doing in the lab in the pre-COVID era, I should say, we also started to ask a critical question here, because most of the work that I showed you so far with the LMNA-mutant disease modeling platform, both in 2D and in 3D, has been done using our transgene-based iPS differentiation protocol. And in the field, there's been around a question for a while, which is, are these patients, and is this disease characterized also by defective myogenesis? In other words, is there a defective differentiation of the muscle progenitors in these patients that might account for the severity of the disease? And so to answer this question, we went back and particularly Dan Moore in the lab and together with Luca went back and took all the iPS lines that we had from these patients. You see 3 examples now, but actually we also have a 4th one, which I highlighted in red here, a new patient that I don't have the time to detail in terms of what we're doing with this other project, and subjected to the protocol developed by Caron et al., which is completely a small molecule based. And you see that we can see in all of them a nice uniform distribution of MI5, which is another key transcription factor regulating myogenesis, telling us that we were in the right lineage. And if you go downstream and look at the final output, again, this is just to test this specific hypothesis. The final output, which is the presence of myosin heavy chain, we see that with a bit of variability, but with no statistical significance, we basically see myosin heavy chain in all the 4 now laminopathy patients and the control without much difference, telling us that there is no major impact of dysfunctional lamin AC on the skeletal myogenic differentiation program, and even more suggesting even more that investing on modeling nuclear abnormality might be the right thing to do, because looking at the phenotype and looking at the actual myogenic differentiation might not be a critical output to analyze. And actually, this is also very relatively fresh data. If you then look closer to this differentiation, to this nuclei in the differentiated myotubes using the transgene-free protocol, you can already see that in 2D, without even putting them in 3D, we start seeing quite a lot of abnormal elongation of those cells. And this is now work that we are trying to complete in the lab, looking at also different options of drug screening to hopefully see normalization of this abnormal nuclear morphology. So in summary, and apologies for having spoken a bit too much, I showed you that this 3D platform that we have developed in the lab It's quite amenable to complex disease modeling, and it's high fidelity in terms of modeling in this specific case of the muscle laminopathies, muscle diseases. And the beauty of the system, in my opinion, is also that it's quite flexible in terms of making it kind of, I should say, it's modular. You know, you can make muscles which are completely made by skeletal muscle cells. You can make bilineage constructs which have muscle cells and endothelial, depending on what you endothelial cells, depending on what you want to study. Or you can have muscle endothelium, pericytes, neuroprogenitors, and we're also now adding other cell types such as, for example, fibroblasts to the cocktail, and could be adapted to a number of different differentiation platforms to derive iPS cells. And I showed you examples to derive muscle cells from iPS cells. I showed you two different protocols, ours and another one in this presentation. So we have a number of projects going on with a number of collaboration looking at applications in regenerative medicine, disease modeling and drug development and gene therapy. For obvious reason, I won't have the time to speak much more about them. And yes, this is the last slide, probably the most important one. So I should really acknowledge the people who did most of the work in the lab. These are the current lab members. Work on the 3D platform has been started in the lab by Sara Maffiretti, as I mentioned, and then completed by Shilpita. And the laminopathy project was started by Heber and then continued by Luca Pinton and Daniel Moore. And more recently also Noreen Kokaras joined the team of the laminopathy crew, I should say, in the lab, looking at different aspects of these diseases. And Kasia is also another visiting postdoc who's helping us in this domain. I should quickly just acknowledge the help of the collaborators in this project. Pete Zammit has been a key collaborator in the laminopathy project. The CureCMD and Cellular Dynamics have given us precious cells, and Giselle Bon, Jean-Marie Cusset as well, Thomas Eschenhagen and Heinz Redlin and Andy Terschlin for the biomaterials, Michael Duchenne and Wilker Tiadis for some physiology measures, as well as Nick Wells at the Royal Veterinary College, and Ricky Patani for the motor neurons. And funding-wise, I should really thank the ERC for supporting this project, but also the NIHR, Muscular Dystrophy UK, BBSRC, and Duchenne Parent Project, who are currently funding the research on this platform in the lab. Thank you very much for your attention, and I'll be happy to answer the questions, I believe, at the end of the session.